Ribociclib, from Phase 1 dose definition to adjuvant positioning, clinical trajectory, and the transparency dilemma, Sep 10th. Note, this article is a correlated and deeper analysis of the piece published yesterday at BBIU, Adjuvant CD4/6 inhibitors in early breast cancer, benefit, cost, and the emerging paradox of resistant relapses. Key findings. Phase 1 defined dose and safety. Ribociclib's recommended Phase 2 dose, RP2D, was set at 600 milligrams 3 weeks on, 1 week off, balancing efficacy with hematologic toxicity and QTc prolongation, NCT 0123736. Phase 2 confirmed breadth, optimization, 400 versus 600 milligrams, equivalence to chemotherapy in luminal B, CORALINE, superiority in aggressive premenopausal disease, right choice, benefit after progression, Trinity 1 maintain, and activity in ER plus ovarian endometrial cancer. Phase 3 MONALEESA program. Across MONALEESA 2, -3, and 7, ribociclib achieved consistent overall survival benefit, HR approximately 0.72 to 0.76, all p.01, with neutropenia as the dominant toxicity. NATALEE expanded scope, first CDK4/6 inhibitor to show invasive disease-free survival benefit in early HR plus HER2 mice breast cancer across both intermediate and high-risk groups at a reduced 400 milligrams dose for 3 years. Transparency dilemma. Resistance signals were observed early, FELINE trial, preprints, yet underrepresented in publications. The credibility of adjuvant CDK4/6 therapy will hinge not only on hazard ratios, but on data transparency and inclusion of resistant patient outcomes. Pharmacological basis. Ribociclib is an orally available, selective CD4/6 inhibitor. Its mechanism of action is centered on preventing phosphorylation of the retinoblastoma protein, RB, thereby keeping the cell cycle arrested in G1. It is metabolized primarily via CYP3A4, with toxicities reflecting its pharmacology, bone marrow suppression, liver enzyme elevations, and QTc prolongation. From its first-in-human studies in solid tumors, ribociclib quickly advanced into pivotal breast cancer programs, ultimately reshaping the therapeutic landscape of HR plus HER2 mice disease. Phase 1 foundation. In the trial by Infante et al., Clin Cancer Res 2016, NCT 0123736, 132 patients with advanced solid tumors received ribociclib. 31% achieved stable disease, and among them 8 patients maintained disease control for at least 6 months, classified as durable clinical benefit. These cases were predominantly seen in liposarcoma and in ER-positive breast cancer with CCND1 amplification. However, the exact number of breast cancer patients in this subgroup was not specified, only noted qualitatively. The maximum tolerated dose was established at 900 milligrams daily, but the recommended Phase 2 dose was set at 600 milligrams, given on a 3 weeks on, 1 week off schedule. Above 600 milligrams, hematologic toxicities and QTc prolongation became prohibitive. Adverse events included grade 3 to 4 neutropenia in 27%, leukopenia in 17%, and thrombocytopenia in 9%. Non-hematologic events such as fatigue, nausea, and vomiting were usually low-grade, while transaminase elevations and QTc prolongation occurred in a minority of patients, but required careful monitoring. Phase 2 development. The Phase 2 program pursued 4 distinct goals: confirming dose, validating efficacy, testing strategies after resistance, and exploring activity in other hormone-sensitive tumors. First, a dose optimization study, NCT 0382248, compared ribociclib at 600 milligrams versus 400 milligrams in combination with aromatase inhibitors in advanced HR plus HER2 mice breast cancer. The trial confirmed that 400 milligrams could maintain efficacy while reducing neutropenia and QTc prolongation, setting the stage for later adjuvant use. Second, validation in breast cancer included CORALINE, NCT 0324842T7, which compared ribociclib plus letrozole against chemotherapy in early luminal B tumors. The primary endpoint, conversion to PAM50 low-risk status, was achieved in 47% of both arms, demonstrating that endocrine therapy plus CDK4/6 inhibition could match chemotherapy in molecular downstaging. Right choice, NCT 0383297, extended this concept to premenopausal women with aggressive disease, showing that ribociclib plus endocrine therapy and ovarian suppression provided superior progression-free survival with better tolerability than chemotherapy. Third, strategies after resistance were tested. Trinity 1, NCT 02732219, combined ribociclib with exemestane and everolimus in patients who had already progressed on CDK4/6 inhibitors, achieving a clinical benefit rate of 41% and a median PFS of 5.7 months. Maintain NC 05207779 explored continuation of ribociclib with a switch in endocrine partner after progression and demonstrated a median PFS of 5.29 months versus 2.76 months with endocrine therapy alone, a statistically significant advantage. Fourth, the program explored other tumors. In NCT 265728, ribociclib combined with letrozole showed activity in recurrent ovarian and endometrial cancers, particularly in low-grade subgroups, where progression-free survival at 12 weeks was reached. Phase 3 pivotal trials. The MONALEESA program established ribociclib as a standard in advanced HR plus HER2- breast cancer. MONALEESA 2 tested postmenopausal women with letrozole, achieving a PFS hazard ratio of 0.57 and an OS hazard ratio of 0.76, with p.08. MONALEESA 3 enrolled postmenopausal women treated with fulvestrant in first or second line, showing a PFS hazard ratio of 0.59 and OS hazard ratio of 0.73, with p equals 0.0455. MONALEESA 7 extended these findings to premenopausal women receiving endocrine therapy and goserelin, yielding a PFS hazard ratio of 0.55 and an OS hazard ratio of 0.76 with 0.0097. Across all trials, neutropenia was the dominant grade 3-4 toxicity, consistently affecting more than half of treated patients, alongside transaminase elevations and QTc prolongation at lower frequency. NATALEE, adjuvant expansion. The NATALEE trial, NC370134, brought ribociclib into the adjuvant setting, enrolling approximately 5,100 patients with stage 2-3 HR plus HER2- disease, both pre- and postmenopausal. The study tested ribociclib at 400 milligrams on a 3 weeks on, 1 week off schedule for 36 months in combination with standard endocrine therapy. The primary endpoint, invasive disease-free survival, was met, 90.7% at 3 years versus 87.6% for endocrine therapy alone, with a hazard ratio of 0.75 and p.005. Toxicity reflected the class profile, with neutropenia in 44% at grade 3-4, hepatic enzyme elevations in 7-8%, and QTc prolongation in fewer than 2%. Treatment discontinuation reached 20%, highlighting the challenge of maintaining therapy over 3 years. Importantly, NATALEE extended ribociclib use to a broader risk population than its competitor. Comparison with MONARCH-E. In parallel, abemaciclib was evaluated in the MONARCH-E trial, NCT 3155997. This study included more than 5,600 patients but restricted enrollment to high-risk subgroups defined by nodal status and pathological features. Treatment consisted of continuous abemaciclib 150 mg twice daily for 24 months with endocrine therapy. The trial demonstrated a stronger relative IDFS benefit with a hazard ratio of 0.66 and p.01, corresponding to a 7% absolute gain at 4 years. However, gastrointestinal toxicity was prominent, diarrhea in over 80% of patients with grade 3 and up to 10%, leading to discontinuations in around 17%. Overall survival data remain immature. By contrast, ribociclib in NATALEE showed a modest absolute gain of about 3% at 3 years, but with broader applicability across intermediate and high-risk patients. Ribociclib also carries the strength of prior MONALEESA trials, where consistent overall survival benefits were documented, something not yet achieved with abemaciclib. BBIU synthesis. The development of ribociclib illustrates a complete translational arc, from phase 1 dose finding through phase 2 exploration and optimization to phase 3 pivotal trials with survival gains, and finally to adjuvant validation in NATALEE. Its advantages are clear: an established overall survival benefit in advanced disease, broader applicability in early disease, and a tolerability profile dominated by predictable hematologic toxicity. Yet the limitations remain equally significant: modest absolute IDFS benefit, discontinuations in 1/5 of patients, and unresolved questions on long-term survival and relapse biology. When compared with abemaciclib, the narrative is strategic rather than absolute. Ribociclib positions itself as the broad-spectrum option with proven survival legacy, while Ribociclib remains the intensive option for very high-risk disease, with heavier toxicity but stronger relative IDFS reduction. The paradox persists: more benefit for fewer relapses, but with costs, toxicities, and potentially more aggressive resistant recurrences. BBIU opinion: transparency, resistance, and the patient's voice. One dimension often overlooked in the current enthusiasm for adjuvant CDK4/6 inhibitors is the question of transparency in reporting resistance phenomena. While clinical publications frequently describe acquired resistance as emerging, patients and independent observers point out that signals of resistance were already visible years ago. The FELINE safety trial, PMID 3471299, documented convergent clonal evolution under ribociclib plus endocrine therapy, a pattern consistent with therapy-driven selection. Further, preprint data from Khan et al. bioRxiv 2021 suggested tumor size increases in ribociclib arms that were later absent from the published version. Observers have argued that this selective reporting downplays resistance and excludes those most affected. They also highlight the pending overall survival readout from FELINE, last patient last visit in 2023, and note concerns that critical genomic evidence of resistance may remain unpublished. From the BBIU perspective, these claims illustrate a structural dilemma: transparency gap. When early signals of resistance are not integrated into regulatory or clinical narratives, the result is a delayed recognition of biological limits. Patient exclusion. If those who experience severe toxicity or resistance are systematically underrepresented in final publications, the evidence base becomes skewed. Structural consequence. What appears clinically as a novel paradox may in fact be the late acknowledgement of a known pattern, suppressed by narrative control rather than scientific uncertainty. For us, the patient's testimony is not anecdotal noise, but part of the epistemic record. The credibility of adjuvant CDK4/6 therapy will depend not only on hazard ratios and p-values, but also on whether data transparency can match the scale of investment and the ethical weight of long-term treatment.
Welcome to the Biopharma Business Intelligence Unit, where symbolic integrity meets strategic clarity. At BBIU, we go beyond the headlines to decode the structural forces behind global biopharma, AI, and economic realignment. If you value precision, verification, and symbolic depth, this channel is for you. Subscribe now to access our weekly briefings, dossiers, and intelligence reports, and consider joining our institutional tier to unlock early warning systems, custom metrics, and exclusive strategic insight. You're not just listening, you're entering the future of epistemic intelligence. www.biopharmabusinessintelligenceunit.com